Hello, I'm Jana.
And I'm Ty. We're in a group.
Okay, so to start with question 1 for today, it gives you a cross and it asks you what proportion have what genotype. So the first thing I did is, as you can see, I drew out all the Punnett squares, and then I just took what genotype it was asking for and just times the number of times it shows up to get the answer.
Cool. Oh, what did you get for that?
So the first one is 1/2 by 1/4 by 1/4 by 1/2 by 1/2, which gives you 1/28, which gives you the genotype capital A, lowercase a, capital B, lowercase c, capital D, lowercase e.
And then do you just want to do all of one and all the
Sure.
other two?
So B is 1/4 by 1/4 by 1/4 by 1/2 by 1/2, which gives you 100 or 256 for the genome. C, we're asking for capital A, capital B, lowercase b, lowercase c, lowercase d, capital E, lowercase e. There is no double or so that one is 0. No genotypes for that because it's not possible. So, and then D is wanting you to do capital A, capital B, lowercase, capital C, capital D, lowercase d, capital E, lowercase e. So that is all by 1/2 because each of them are 50%, and that gives you 1/32 possibilities of that being your option.
So I'm going to go over like the vocab. Can I? Yeah. Cool. So a pair of homologous chromosomes are often sometimes called tetrads or bivalents. The structure that connects the centromere to the microtubules of the mitotic spindle is called a kinetochore microtubule. A diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 16 sister chromatids, and then while fruit fly cell in metaphase 2 has 0 sister chromatids. So we know that those are only present in meiosis 1. A null allele is a type of genetic cross that can determine if a purple flower, a monohybrid cross is a type of genetic cross that can determine if a purple pea plant, and then determining the genotype. A non-functional allele is sometimes called a null allele, while a partially functional allele is called a hypomorphic allele. Mendel's law of segregation involves 2 traits, while Mendel's law of independent assortment involves a single trait.
So for number 3, we did not understand what's asking.
We really struggled with that one.
Because it's asking for your possible homologous chromosomes, but we're talking about alleles, so you don't know where on the chromosome that allele is or what chromosome. So
okay, and so then for 4, I have, let's see here, how many chromosomes are present per horse cell at the conclusion of meiosis 1? So we know that 2n equals 64 and n equals 32, and so A would be 128 chromosomes. And then how many tetrads are present in meiosis 1 would be 32. How many sister chromatids are present would be 64, and that's in meiosis 1, but when it asks the next question in meiosis 2, we know it's going to have 0 sister chromatids because they separate. And then it says how many, it would be 0, they separate. And then it says how many bivalents are present in anaphase 2, and that's going to be 0 too because those don't exist in meiosis 2.
So number 5 is a lot like number 1. We're doing a Ti-hybrid cross, and it gives you your pea plant alleles. And so I wrote out again all of my Punnett squares, and then ask you which amount will be tall, round, green, and terminal. So you look up the amount that would have each one of those and times them together, and you get 9/64. be asked which one will have the genotype little t, large r, little r, capital Y, lowercase y, and capital A, lowercase a, and that is 1/32. C is how many of the offspring will be heterozygous for all 4 genes, and that is 1/64. And D is what offspring will be dwarf, wrinkled, yellow, and axle, and that is 1/64.
Oh wait, and then C is 1/16, right? That's what.
Oh yeah, that's 1/16. Sorry, did I say that?
No. Awesome. That's our video.